Caloric restriction has emerged as one of the most consistently effective interventions for extending lifespan and improving health span across a wide range of species, from yeast and worms to rodents and primates. One of the primary reasons behind this phenomenon is its ability to activate autophagy, a process that allows cells to clean themselves from within, conserve energy, and adapt to stress. Rather than being a mere response to starvation, autophagy is a highly evolved survival mechanism that improves cellular resilience and promotes long-term balance. When food is scarce, instead of descending into chaos, the body shifts into a protective mode. It stops growing and starts repairing. At the heart of this shift is a subtle metabolic reprogramming. In the fed state, when glucose and amino acids are abundant, the cell prioritizes growth, protein synthesis, and energy storage. But during caloric restriction or fasting, nutrient-sensing pathways like insulin/IGF-1 and mTOR, mechanistic target of rapamycin, become downregulated. These signals, which ordinarily promote cell proliferation and anabolic processes, give way to catabolic pathways, particularly autophagy. Once triggered, autophagy begins the systematic breakdown of old, damaged, or redundant cellular components, converting them into usable building blocks like amino acids and fatty acids. These recycled elements can then be used for energy or as raw material for essential repairs, all while reducing oxidative stress and cellular clutter. The longevity benefits of this adaptive cleanup process have been demonstrated repeatedly in laboratory models. In mice, caloric restriction not only extends lifespan but delays the onset of age-related diseases such as cancer, neurodegeneration, and cardiovascular dysfunction. In fruit flies and nematodes, the effect is even more dramatic, often increasing lifespan by 30 to 50%. Importantly, when autophagy genes are silenced in these organisms, the longevity benefits of caloric restriction disappear, strongly suggesting that autophagy is not just a side effect but a central driver of the aging delay. Beyond lifespan, caloric restriction-induced autophagy improves the quality of aging by preserving organ function. In the brain, autophagy reduces the accumulation of misfolded proteins and helps neurons maintain metabolic efficiency, potentially lowering the risk of Alzheimer's and other cognitive disorders. In the liver, it helps detoxify cells and regenerate tissue. In the heart, it reduces inflammation and prevents fibrosis. Even in the gut, autophagy maintains the integrity of the epithelial barrier and supports a more balanced microbiome. These benefits are not limited to long-term fasting. Even intermittent energy restriction, such as time-restricted feeding or alternate-day fasting, can prompt periodic autophagic pulses that help refresh and rejuvenate tissues. One of the more encouraging discoveries is that cells use autophagy not just to survive restriction but to thrive through it. By removing worn-out mitochondria via mitophagy, cells can stimulate the birth of new, more efficient ones, a process called mitochondrial biogenesis. This renewal leads to better energy production with fewer free radicals, which in turn reduces DNA damage and slows the aging of the genome. The entire process is a virtuous cycle. As autophagy clears damaged parts, the cell becomes more energy efficient, stress-resistant, and capable of functioning under changing conditions. Caloric restriction also fosters a deeper kind of metabolic flexibility, the ability to switch between fuel sources and cope with fluctuating nutrient availability. This flexibility is not only a marker of good health, but also a predictor of longevity. Autophagy supports this by adjusting the balance of protein turnover, fat oxidation, and glucose use, ensuring that the body can adapt without incurring metabolic damage. It also works in concert with other protective pathways, such as sirtuins and AMPK, all of which are activated during energy stress and contribute to anti-aging effects at the cellular level. The beauty of this system is that it's deeply accessible. While severe, lifelong caloric restriction may not be practical or even safe for most humans, milder strategies like intermittent fasting, time-restricted eating, or simply reducing calorie intake by 10 to 20% can offer many of the same benefits. These approaches stimulate autophagy intermittently, which appears to be enough to maintain a healthy baseline of cellular renewal. Moreover, caloric restriction mimetics, natural or synthetic compounds like spermidine, resveratrol, and metformin, are being studied for their ability to trigger autophagy pharmacologically, potentially offering a way to reap the benefits of fasting without reducing food intake drastically. In summary, caloric restriction activates a powerful self-preservation program that turns down growth, turns up repair, and helps the body economize its resources in a way that promotes longevity. Autophagy is at the core of this transformation. By clearing away what no longer serves the cell and recycling it for a higher purpose, the body enters a mode of intelligent conservation and strategic renewal. Aging becomes less about inevitable decline and more about continuous adaptation. When we understand this, caloric restriction is no longer just about eating less. It becomes a tool for cellular wisdom, resilience, and sustainable vitality.